Earlier today we had, thank you. Earlier today we had the plant ecology, Dryland plant ecology session, and now we continue. This part of the session will be dedicated more to interactions in plant ecology, and we will have the majority of the presenters from the group of my friend here, hello, which we will learn about his team as well. So the Finkel lab is leading, and let's start the session and enjoy.
Thank you, Tamir. Hello, everybody. So we have a big room, but small, intimate session, and many of the people in the audience are from my group. So people that are not from my group, it's nice to meet you, and happy you came to listen to us in this late hour. So this is weird. I have Noah's presentation on this computer, but that presentation on the screen, I guess. It will move, yeah. All right. So since the organizers were kind enough to give two people from my lab a talk in this session, and I don't want them to overshadow me, I will pivot and talk about something a little bit different than the original title. I will talk about a separate project. I'm going to talk about nodules. But first, just a quick introduction about what I was supposed to talk about, and happily Noah and Tingting will talk about. So there's the big project in our lab that is basically trying to ask one big question, which is, did plants adapt to recruit beneficial microbes when they are under drought in order to deal with this drought? And a smaller question, which is, how can we even, if this is true, how can we even prove this? And we designed this scheme where we're using the ecology of plants across Israel and comparing closely related plants that are found either in the north or in the south of the country to try to answer this question. And like I said, you guys can stay tuned a little bit later and hear more about this, but I'm going to pivot to something completely different. Well, not completely, but to a different project. And I want to talk today about the project that this guy here, Dr. Moshe Alon, he's a postdoc in my lab, and he brought this project to my lab. It's actually something he started as a PhD in Dr. Efrat Sheffer's lab, and it evolved into a really, really fun collaboration. So this is what I want to talk about today. And Moshe's favorite plant is Calicotoma viosa. It's a legume shrub, Kida seira. It's found all across Israel in many different places, and this is Moshe here sampling some Kida seira in the wild, and this is the first thing he did in his, and he was in Efrat, it's a plant ecology lab, and he did a very, very major survey of different populations of Calicotome, where they can be found, what types of soils, what type of climates, the size of the population, and so forth and so on. And one thing that they noticed, which left them scratching their heads a little bit, was that this site over here that they sampled, Adulam, which climatically should have it's sort of a Mediterranean shrubland. It should contain Calicotome, but it doesn't. They couldn't find even a single individual from this species. And this was a bit weird for them. And since it's a legume, it led them to think that maybe this is related with the symbionts. And Moshe came to my lab with this idea of trying to figure this out. And I came on board, and Moshe is still in my lab now working on related subjects. So yeah, this is another, right, this isn't Moshe's survey, this is just from BioGIS. And you can see, again, there's sort of a hole in the distribution of this plant, not only in his data, but in cumulative public data. So the first thing I asked Moshe to do was to isolate the symbiont from places where the plant is growing and growing happily. And after working a lot with microbiome studies on different plant species, this was really fun because you know, when you're working with this legume symbiosis, you have a large population of a single bacterium. You can isolate it from these legumes rather easily. And he was able to do this even though he had very little experience as a microbiologist. So here we have an isolate from a site in Ramat HaNadiv up north where there's many Chalicotome, and we sequence its genome. And you can see it's right here. This is a tree of many related it's a Bradyrhizobium. Chalicotome forms the symbiosis with Bradyrhizobium. And here you have our friend here, RHA1, sort of in the middle of the tree. And we have a bunch of different traits here. You see, it's kind of difficult to find correlations between different traits, but all of these related species on this branch of the big tree, you see this is a magnification of this branch. These are all Bradyrhizobium. You see all of them tend to be from Mediterranean regions, not all. Most of them are nodulating. They come from many different plant species, some from acacia, some from populus. So it seems like even closely related species will switch hosts and form symbiosis with different hosts. But anyway, this is the symbiont that we found. And basically Moshe did a very simple experiment. He grew the kida this is the Ramat HaNadiv soil. This is where the symbiont comes from. You don't need the symbiont. This is with the symbiont. This is without. The plant is very happy. But if he grows it in this adulam soil, the kida isn't able to grow. It very barely. It doesn't form any nodules, and it eventually dies. But if he adds the symbiont that he found here, the plant is absolutely fine, and very similar to the way it grows in Ramat HaNadiv. So this led us to conclude that the distribution of the plant isn't only driven by abiotic factors and the climate and the soil type and so forth, but also by the distribution of its symbiont. So this is that's where he very happy he published this paper, and we moved on to study a few different things. And one thing we wanted to do was, since we were looking at, we were isolating bacteria from these nodules, we wanted to know, well, when we isolate bacteria from the environment, we're introducing a very strong bias. So we don't even know if this particular isolate that we picked up is necessarily the dominant species in these nodules. It could be just one that is preferred, that grows better on the media that we've used, and we wanted to have an unbiased way to look at what the different populations of the symbionts look inside these different nodules. So again, we went back to Efrat's lab, and they've did many surveys, and this is actually nodules that were sitting for quite a long time and dried up in envelopes, and we were able to extract DNA from them and send them to shotgun metagenomic sequencing. And these are some of the results. So this is all, if you're familiar with microbial ecology, you know that many studies use 16S amplicon sequencing, which is great. It gives you a quick picture of what the community structure looks like. But here we didn't do any of that at all. We did only shotgun sequencing, which means you sequence everything that's in there. This is quite risky with endophytes because we risk having a lot of plant DNA just interfering with our sample. But happily, this wasn't the case. And once you get a quality sample of metagenomics, it's really much richer than just the 16S. You can do many things. And also at the taxonomic level, I happen to believe that you get a more reliable picture of what's happening. And here we see, this is at the genus level. This is using a new tool called Single-M, great tool to extract taxonomic profiles from metagenomic data. And you can see, as expected, that at the genus level, most of these 5 different sites, so it's 5 plants from each site, 5 different sites from different parts of the country. This is, again, RH is Ramat HaNadiv. That's the same site where the original isolate came from. This is Ramat HaNadiv. There are 2 sites in the Carmel, Caesarea, Chokuk, Metzel. And you can see that most of the nodule is, as expected, Bradyrhizobium. But then some sites have more, like especially this site in Metzel, has a higher diversity within the nodule, and these are surface sterilized nodules. So these are endophytes that were found within the nodule. We find out also a lot of other stuff, and this raises the question: what is this stuff? What are these bacteria doing? But as for this Bradyrhizobium, we saw previously that there's quite a diversity of this genus. Are these really all the same? And within each plant, do we see the same, just a monoclonal population of the same symbiont? So the answer is no. It's enough to go to the species level. You can see these are all different colors. All of these different colors are different species of Bradyrhizobium. And you see there's actually very high taxonomic diversity within the genus. So the Calicotome is forming symbiosis with very different Bradyrhizobia. Each site has at least 3 or 4 different populations of Bradyrhizobia within the same plant, and different sites have different populations of Bradyrhizobia. And this is an opportunity, right? This is interesting, and it's an opportunity because they're going to have different genomic contents, and we can try to start linking the genomic content of these symbionts with the plant phenotype, the environmental conditions, and try to understand why this ecological pattern is forming. Now, this is all done with unassembled data. So these are just short metagenomic reads that we annotate. But we can also assemble this data and extract genomes. And since, again, these are relatively low diversity samples, that works very well. So here you have these different circles here all represent metagenomic assembled bins. So these are different genomes that we've extracted from the different sites. So you can see we can find each site has a sort of a typical sort of primary genome that's found in, for example, Ramat Nadiv, we have 2 very different genomes from the same site. They're both Bradyrhizobial symbionts, but they're very, very different. And then this here is the isolate that I talked about earlier, the one we used in the previous study. You can see it's relatively close to this genomic bin, but there's still some distance between them. So there's a lot of intergenous diversity in this group. All right, so like I said, we want to understand, don't try to understand anything in the slide, it's not the purpose of this, but we want to understand, can we link, we have all the genomes, we have the functional content of the genome, can we find functions that are found or not found in these genomes that predict how the plant's going to do and its performance? So this is kind of an attempt to do this. Again, don't try to figure it out. Each row here is a different genome. Each column is a different function, and it's sort of linked to different plant phenotypes. But let's just focus on one function that we saw an interesting differences, and that is, right, these are all nitrogen fixers, but it turns out that besides nitrogen fixing, they have another nitrogen cycling trait, which is denitrification, right? So this is basically the opposite of nitrogen fixing. If nitrogen fixing is taking nitrogen into gas and putting it into the soil, denitrification, which is sort of a form of anaerobic respiration, is taking nitrate and turning it back into gas and pushing it out to the atmosphere. This is very beneficial for a symbiont found inside a nodule because it's in a microaerophilic environment, and this provides it with an alternative electron acceptor to oxygen. But we don't really know what this does to the plant. We notice that not all of the symbionts, or not all of the nodules, have symbionts that contain denitrification operons. So, and the main gene is the nitrite reductase, NERK. That's sort of the key gene. That's really the step which turns it from a soluble form, from nitrite to a gaseous form. So this is what the gene we're looking at, and you can see there's a difference here. So we can see, generally speaking, that plants that contained symbionts that had the NERK gene also produced higher nodule biomasses in general, and this seemed to lead to also, in this case, to larger plants, to more plant biomass. So it seems generally that nodules that have NERK across these different sites are doing better, are happier. So that was an interesting observation here. Just again, right? I took this from Wikipedia, right? Nitrogen cycle. So this is basically what I just said. Right. And NERK is this gene over here, from nitrite to nitric oxide. All right. So this was an interesting observation, but this is an observational study. we don't have any sort of hard experimental data. So Moshe did another experiment, and here he shifted from working with calicotome to working with not a perennial but an annual legume with medicago. And again, we want to see if the identity of the symbiont really affects how the plant responds to unavailability, and can it sort of control the size of the of the nodules and eventually the size of the plant itself. So he set up this experiment with medicago. This is done in sort of an inert substrate in perlite, and then he adds different symbionts that he collected from different soils that he isolated. Now he's gotten pretty good at isolating these symbionts. So he collected them from different soils and inoculated the plants with them. And and he did this under two nitrogen concentrations, under high nitrogen fertilization and low nitrogen fertilization. I'm going to take a sip of water, just a second. Okay, much better. So, so these are these 4 isolates that he found. Again, this is one from different sites, so it doesn't matter what they are, but these are all different places across the country. This is Nestiona, not New Zealand. And let's see what we got, right? So here he's looking at the nodule mass fraction. So this is basically, he's taking the mass of the nodules in the plant and dividing it by the total biomass of the plant, and this is kind of tells us something about efficiency, right? Because the lower the nodule mass fraction is, the more efficiently the plant is utilizing its nodules, right? It's getting more biomass from less investment in nodules, and both under the high nitrogen treatment and the low nitrogen treatment, right? The nitrogen is given here as nitrate. We have this NZ site again, Nestiona site, kind of elevated above the others, right? So it has a higher nodule mass fraction, suggesting that again this plant is producing more nodule biomass, but not necessarily getting the benefit from it. It's not getting more plant biomass from this. So we looked at the genomes of these different isolates, and it turns out that this NZ isolate was the only one who is a denitrifier. So we're back to the denitrification again, and here we can see over here that it has this almost a complete denitrification operon. So it goes all the way to nitrous oxide, not all the way as we would like to N2, but it definitely has the denitrification genes. So yeah, this was curious to us. These are already 2 cases where we see that the presence or absence of this nitrogen cycling process have an effect. Not, of course, we're interested in this because this is related with greenhouse gas emissions, but it also has an effect on how the plant responds and interacts with its nodules. Here we have, this is a phylogenetic tree of all of the nodule-lating, nitrogen-fixing bacteria that have been sequenced to date in all of the databases. So it has a bunch of different taxa you see. There's over here is the sort of the weirder ones, the ones that aren't rhizobia, but all of them have NOD genes and NIF genes. And the main sort of circle you want to look at is the one here in between these two. This is NERK again. And you see that it's distributed kind of randomly. There's not a strong phylogenetic signal, but it's found all across the tree in about more or less 50% of the bacteria of these isolates. So this is kind of interesting. It's probably found on a mobile genetic element, usually on a plasmid, and it's sort of gained and lost in evolution, right? So this may be sort of reminiscent of some kind of cheating phenotype where the symbiont is taking away a little bit from the plant, cheating the plant a little bit, but not a lot, right? It's still fixing its nitrogen for it. But it exists in a sort of equilibrium where under certain conditions it's not beneficial to retain this process. So this is quite an interesting thing to look at. But really the way to study this carefully and to link it directly to the NERK is to go to to do some molecular biology and knock out the gene. And after a lot of efforts, Moshe was eventually able to knock out the operon for denitrification. We did this in collaboration with the dual means lab in Vulcani. We measured N2O emissions, and you see this is the wild type emitting a lot of N2O, and here's our knockout bacterium. So it's not denitrifying anymore. It's not emitting anymore N2O. And Moshe is now running some experiments with this, comparing the wild type to the mutant. And it seems like he is able to find some differences. So here you see the wild, again, nodule mass fraction, and you can see in the wild type there's no difference in the nodule mass fraction between the presence or absence of nitrogen. This is also what we saw in the original experiment. But in the mutant, we can see that the plant is able to respond to invest less in nodules when nitrogen is added. So right here, the plant here is more flexible. It's able to get lower, lower nodule mass fraction, to be more efficient when it doesn't need to invest so much in nodules. When it has other nitrogen sources in the soil, it's not able to mount this response under these conditions. And with that, I will conclude. So just to summarize, it looks like denitrification in nodules is relevant not only for the symbiont itself, but for the plant. And if we go out to the field and we sequence these communities and we look at the content of them, we can definitely find some actionable items that we could later on study more in the lab. I want to thank my lab, many of whom are here in the room. Thank our collaborators, Efrat Shefer, Amir Erez, Dr. Dror Mintz, where we did our denitrification essays. And thank you. Thank you for listening. Happy to take questions.
Questions to Omri?
Hi, Omri. Thank you for the talk. So in the last slide, you showed about nodule mass fractions. So it is like affected by the number of nodules, or is just the fraction of the of the?
We don't really have a good quantification of which is the higher driver, but I don't think there's less nodules. It's just generally smaller ones. Yeah.
How specific are these interactions between symbionts and these hosts? Because I was surprised, like it was missing just one Bradyrhizobium species, and that was it.
Yeah. So the question of specificity, I think, is an open question, and there's research about it, and different plants seem to have different levels of specificity. For Chalicotomys, it looks like it only forms symbiosis with Bradyrhizobium, but we see that it also formed them with many different Bradyrhizobia, and sometimes in the same plant in most cases. But yeah, in the case of this particular site, it looks like the entire genus is not, doesn't thrive very well in this site, and that might be the reason. We don't know why Bradyrhizobium doesn't like Hadulam, but yeah, that will be a topic for another study.
Nowadays, we are talking a lot about functionality matters a lot rather than the taxonomy. So, here it's not following the criteria, or what's really going on? Because we are talking about the function is more important for the microbes. What kind of function they can do for the plant or for the host is more important rather than the taxonomical categorization that they have.
We know that there is a link between taxonomy and function. For us as researchers, if you can get data that gives you both the taxonomy and the function, then it's richer data, right? And then you can find the places where they depart, where you'll have closely related species that have a different genomic content, and that'll explain things that you just can't see if you only look at the 16S.
Thank you, Omri. So next up, Assaf. Yeah.
All right. Hi, everybody. So I'm just finishing my master's at Omri's lab, which you've just heard from. And today I'm going to talk to you about harnessing microbial partnerships, drought resistance in desert-adapted plants. So, as you've probably heard a few times by now, even only today, with the growing levels of CO2 in the atmosphere, we're experiencing expanding trends of desertification. Meanwhile, the growing population is by itself creating a real concern regarding food security. And when you combine these two factors together, so it really brings us to a place where we really need ways to grow food using less water. What I'm going to show you, what I'm going to talk to you about today, is a way in which plants might be already doing that naturally. So, it is known that plants release from the roots to the soil a combination of compounds. And that combination of compounds cultivates a specific microbiome. Now, the cry for help hypothesis says that when a plant is experiencing stress, this stress might make it change what compounds it secretes into the soil in a way that will change its microbiome and help it deal with the stress better. Now, this was studied mostly in biotic stress, like, for example, a caterpillar eating a plant. But in our research, we were trying to look at abiotic stress, and specifically drought stress. So we were trying to find if when desert plants are experiencing drought, if they change the profile of exudates in a way that recruit bacteria that help them deal with this drought stress. So, how did I try to check it? So, I was working with two local Israeli plant species, the Mediterranean Isatis lusitanica and the desert Isatis microcarpa. Now, these plants are very closely phylogenetically related, but they have a different natural distribution, as you can see in the map, in different climate areas. So, actually, the desert Isatis microcarpa has much more bigger stress on it to develop drought resistance mechanisms. So, we're expecting to find this mechanism that we're looking for either only in this desert species or much strongly in this desert species in compared to the Mediterranean one. So we took these two species and we grew them in the lab in syringes filled with sand. And also I left some syringes without plants as control. Half of the plants received wet irrigation, which received wet treatment, which meant regular irrigation, while the other half received dry treatment, which meant no more irrigation since the beginning of the treatments. And after 7 days, I collected the exudates by dripping water from the upper side of the syringe, collecting what came out on the other side, instantly freezing it and analyzing it. So, what did I see? Well, in my results, I'm going to refer to 3 different components. I'm going to talk about the plant's drought tolerance. I'm going to talk about the plant's root exudates, which, to remind you, is the compounds that the plant is secreting from the roots to the soil. And I'm going to talk about the microbial community. So let's start with the drought tolerance. Just one more thing to say is that these plants that we were working with, they're not model plants. We didn't know almost nothing about them starting out, and we also didn't really know if they had any drought tolerance, any difference in drought tolerance between them. But just by looking at the plants at the end of the experiment, You can see that the desert Isatis microcarpa seems to be handling the drought better than the Mediterranean Isatis lusitanica. But let's look at some numbers. So first we wanted to look at the biomass. So if you're looking at the root biomass, so you can see that we didn't see any change in either of the species between the two treatments. They didn't change the root biomass under drought. However, when we look at the shoot biomass, so the desert species again didn't change its biomass, but the Mediterranean species had a significant decrease in shoot biomass under drought, which presents a very classic response to drought. The plant has experienced drought, and so it invests more in root biomass to look for water. The next thing I looked at was the stomata opening of the plants. And again, in the Mediterranean species, we see a very classic response to drought. The plant has less water, and so it closes the stomata to conserve water. However, the desert species, again, no difference between the two treatments. And then I tried to look at the photosynthetic activity of the plants through electron transport rate. And once again, in the Mediterranean species, I saw basically what we expected. There was a decrease in photosynthetic activity under drought. It has less water. Now it also has less CO2 since it closed the stomata. So it's like very what we expected to see. And again, the desert species, no difference between the treatments. And also we can see that it keeps around the same electron transport rate as the Mediterranean species under regular irrigation. So generally, our desert species seems to be doing quite well under drought. So now let's look at the plant's exudates. First of all, I wanted to look at the amount of total organic carbon. And you can see here that when we look at the Mediterranean species, there is no difference between the two treatments in the total organic carbon. But when we look at the desert species, it seems that it's secreting more organic carbon to the soil when it is under drought. The next thing I wanted to look at was the entire profiles of exudates, not just amount. Now we detected more than 1,000 different compounds in the samples. And here you can see like all of them here together in a PCA. And what's very clear is that the two controls, I'll remind you, it's the syringes that didn't have plants inside, but still got either wet or dry treatment. And you can see them clustered separately here on the side. So we can say that the difference between the samples did not come from just the fact that the sand was either wet or dry, but from the existence of the plants inside. And when we look just at the plant samples, so we can see a very, very clear separation to different groups. So you can see here in light green the regular irrigation of the Mediterranean species, and here the drought treatment of the Mediterranean species. Here we have the regular irrigation of the desert species, and here the drought treatment of the desert species. And apart from seeing this very clear separation, you can also see that they have generally the same response to the drought. But I want to look at it in another way. And here I was looking at the number of compounds that were either enriched or depleted under drought in each of the two species. And you can see that there are much more compounds either enriched or depleted in the desert species. So it has like a much more stronger response to the drought. But does this stronger response in exudates correlates to any change in the microbial community? Well, the answer seems to be yes. So here you can see the results of 16S sequencing that Omri mentioned before. And you can see that only in the desert species, it's like a 16S sequencing of the roots of the plant. And you can see that only the desert species has a significant difference in its bacterial community under drought. Well, so what did we see so far? We saw that the Isatis microcarpa, the desert species, unlike the Mediterranean Isatis microcarpa, keeps its biomass allocation, its stomata opening, and its photosynthetic activity under drought. It also has a stronger exudation response to the drought, and it has a significant shift in the bacterial community. So we can say that it definitely looks like this plant has drought tolerance that is related to the exudates and to the microbial community. But we still have a few pieces of the puzzle missing. So what we're planning to do next, we're going to look at individual compounds. We're going to look at functions rather than strains in metagenomics. You're going to hear about it soon. And we're going to look at more plant pairs. Again, like Omri mentioned, this was just one plant pair, the Isatis. And also, my part of the project was focused on the exudation parts. But Tingting, which you're just going to hear from after me, is doing a much more comprehensive work on the microbial community. And lastly, here I showed you a difference in the drought tolerance of the plant, and I show you a difference in exudates and a difference in each one of these three components. But we want to make sure that there is not only a correlation between them, but also a causation. So in the next experiments in the lab, we're gonna test exactly the relationship and the connection between each one of these components. Thank you very much to Omri, my supervisor, and of course to my amazing lab. Couldn't have done it without you. If you like this project and would like to take part of it, since I'm leaving, so feel very welcome to talk to Omri about it. And that's it. Thank you very much for listening.
Don't be leaving just yet. Don't be leaving just yet. Okay, good. One or two questions for now. So I'll start. Do you think that you could later on, or Omri, connect between specific metabolites and specific bacteria?
Well, I hope so, but obviously we won't know until we test it. But that's one of the things we're going to do next. I think Tingting also planned a specific experiment for it. So we'll see.
I think this work is fascinating, and we should talk more. Yeah. So more questions from you guys. They're just dazzled. They're overwhelmed. Okay. Thank you, Noah. And Tingting, you're up next.
Okay, thank you everyone to come to the Finkel lab meeting. I'm the next step of Noah, so I'm continue to do this work. It's a big project from our lab. So today I'm gonna talk identification of microbe species and genes associated with drought tolerance in desert Brassicaceae. So 1 gram of soil contains 10 to the 11 microbe cells, and we are interested in how these microbes infect the organism around them, and specifically plants. So from the plant point of view, so the microbes range from beneficial to neutral to pathogenic. And together of them to form a plant-associated microbiota. So which this community distribute in different plant issues, and also the rhizosphere, the surround zone. So basically, such as like philosphere and rhizosphere and endosphere. For our work, we are more focused on the roots. So basically, is rhizosphere. So we knew the microbes contribute to the plant nutrition and development and defense and stress tolerance, and the plant physiological response can reshape the compositions of communities. From this point of view, we want to ask, like, how plants evolved along with their microbiota to cope with aridity. Okay, so you already see this map from Omri's talk. So to address these questions, we designed experiments with the different species of Brassicaceae, and we start now with 3 species of Brassicaceae. They are distribute different precipitation gradients from the north to the south. So when I mentioned the north species, you can think about it as the plants original from Mediterranean area, and if I mention is a south species or drought species, it means they are original from collected from the desert area. So this provide us a really natural eco-evolutionary framework to study how arid adapted plants to involved to collect specific microbes under drought stress, and also how the recruit the microbes, how they help plants to deal with the drought. So for the soil part, in beginning, we are planning to use 3 kinds of soil. From the work I show you now today is only focused on Jerusalem soil. It's kind of a huge experiment. And also, we will test the particulars of like the soils, and we found out like for Lahavim soil and Stillbrook soil, it's kind of have less nutrition and high salinity. It's kind of hard to grow in like 1,000 plants like from the originally from the south, north. It's kind of hard for them to deal with this stressful in the beginning. So we are continue work with the Jerusalem soil as like the base. So for the experiment setup, we have 6 species and Jerusalem soil, and we have treatment of white and drought. In total, we have 60 pods to do that. Oh, this emoji is wrong. So basically, in all of the like drought treatment, people always mentioned they are give the treatment to drought, but it only stop, the way they do is like stop watering them after a week or 2 weeks, then they collect the samples. It's kind of like hard to work with different soil and different plant species because the transpiration things and the properties is kind of different. So it's hard to do quantity of them, and also kind of hard to repeat your experiment. So for us, we are using the sensors to test the water content and the water potential. So each of the time we knew exactly the time point when we harvest the samples, and like for each of after repeat experiment, we use the same conditions. So which in our now the conditions is like we collect the soil, as you see here. X is different days, and Y is water potential. So you can see from here is like the plant growing. We usually move them to the soil after 10 days growing them, and we start the treatment. So you can see, start from the day you give the treatment, the water potential is going down until like around minus 600 to 700 kPa. This is the point we harvest all of the samples. Yes. So this is after the treatment. You can see the phenotype. It's kind of like in the soil system. It's kind of different as what Noah showed you before in the sun system, because in soil is more complicated and you have microbes there. They have interactions. It's so, and even in this southern plant, the northern plant is like a desert. Suppose the desert plants. So you see they are quite similar after the drought. They don't have like significant difference between the phenotype. But as you see, they both like what we want to know is like they are really under the drought treatment. So you can see from the phenotype. So here I give you one example. So we also test all the physiological parameters, so the stomatal conductance and the transpiration rates. As you can see, from the 1,000 species, they are more lower than the northern species. So it means like under the drought, they deal with the drought better than they are like they have lower of like stomatal size, then they can keep the water well. So from this data, you can tell is like the 1,000 plants actually can perform like better to deal with the drought. So next, we want to ask like which component of the root microbiota are drought responsive. So for this reason, we collect the microbes after the treatment, which is from rhizosphere, rhizoplant, and the endosphere. And we sequence them by 16S, ITS, and metagenomics. And also we collect the plant tissues, do transcriptomics. And because of a time issue, I'm not showing you all the data today. So this is the data after we doing metagenomics, and we use the singulum to use the single copy gene to quantify the compositions of each of different plant species, like the microbe compositions. So as you can see in rhizosphere, and after the drought, they didn't have really big shift from the composition from before, like under control. So it's like kind of like already known. Is a lot of other studies also show rhizosphere is not that sensitive to gut like shifting, but once you can see the rhizoplant is like on the root surface we will collect. So you can see there is a strong shift after the drought, which is Pseudomonadoda and Actinomycetoda. So you can see there's a significant change after the drought. So it makes sense because it's the part close to plant roots, and the roots can secrete metabolics and can affect the composition of the roots, the community. So after that, we want to also know like which microbe functions module are associated with the drought stress response. For this point, we are using our metagenomics and directly to match the cake pathway and to see the gene functions. So from here, like from this PCOA, you can see like a root compartment dominates variation, which is means this white dot is from rhizoplane, and this blue from rhizosphere. As you can see, is more like the variation is more between the different compartments. But you see, like in the rhizosphere, you also during the drought and the control conditions, they are shifting the things. So not much in the rhizosphere. Same as the result you see before. So we are matching the, now this is like top 50 cake gene functions. So so you can see here from the top is the control, the green is control and blue is drought. So you can clearly see here the enrichment during the drought in rhizosphere, so which is the same what we say before. So we can tell here like strongly like drought responsive functions is in rhizoplane than rhizosphere, and also we will focus on the functions and to see which like pathway they are belong to. So what we do after we identify the gene functions, so we want to also link the functions to bacterial isolates, and we just got the data not far time ago, so we still managed to assemble the MAG. So after we have the MAG, we can know which this gene from which bacteria, so we can know exactly the species. So we will during this, like busy on this information, so we will build our syncom community experiment to verify their functions. So for this purpose, we need to isolate a lot of strains and to make the syncom. So I'm not going to detail. The multi-top is like use the you can test the biomass, then compare with them. It is a very good method if you already have the library of the strains. So once you put the strain, in 1 second they tell you which strain they are belong to. So you don't need to sequence them, to spend the money and the time. So you can, from a really beginning, from the colony, you already can select them which one to continue work with. So in the end, we managed to have 1,341 isolates from the 4 main phylas. So if you pay attention, so we have like 10 different phyla from the metagenomics data, but in the real life, it's kind of hard to isolate the strain from another 6 different phylas. So we're still working on that. So this is the one we have. So we will build the syncom based on that. So take-home message. So first, the drought strongly enriched Pseudomonas dota and Actinomycetoda in rhizoplane. And the root compartment is the main determinant of root microbiome compositions, and the root compartment and the host identity joins shape the taxonomy and the functional profiles of root microbes under the drought. So the drought response cake functions are concentrated in the rhizoplane. It highlighting is a K plant microbe interface under the drought stress. And I want to thank you, my advisor, Omri Finkel, and I want to thank you all of the lab members from Finkel's lab, and also specifically thank you, Yael is here, and Jordan to work with me, and thank you, everyone.
Thank you, Tingting. We have time for one question. They are not ready yet. You've come from the future. But stay. I want to ask you something. I hope
next time I can show the whole story to you.
So, one question about is it possible that in your analysis you also mix between dead and live bacteria, or are you sure that everything is active, live?
I mean, like I mean, when you collect the samples, actually you cannot tell it's survive because it's like with DNA, so you don't know, yeah. But you know, like the syncom experiment kind of can construct what way we want, and they all alive, so you can see like exactly after you give the syncom, and you see like the shifting and the drought, or like there's a specific recruiting. Yeah.
Are you giving the talk next? Yes. Come. Why should I go?
So it's about the method, like when we go for shotgun metagenomes, and you are extracting it from the roots, and you already said it in the talk that you have a lot of plant DNA inside. So you did some enrichment and like before going for shotgun, or is just extraction?
It depends which part you use, because for us we have a rhizosphere and the rhizosphere and endosphere, so we didn't manage to sequence endosphere because it's so many, but we have the data from 16S and ITS. I didn't show here the time issue. So, but from, you know, like also when you are, it's kind of, they have crucial step. If you really want to avoid a bit of part of tissues, you can filter them before you extract DNA. Yeah, but for us, we were checked the contamination is between like 1% to like 10%, so it's not really high. So we have enough risk, so we just delete it. Yeah.
Thank you. Okay, so last speaker for today.
So I have enough time.
Yeah. Yes. Uttam Kumar from Ben-Gurion, from here. Are you in Sdeboker?
Yeah.
Yeah. Good.
Okay, so thank you for being so patient that you are still here till the last talk. And I can see all my friends, and Yael is here, Professor Simon Barak is here. And thank you all for being here. And they want to see what I have been doing the last 3 years when I'm going to the lab. So we can change it from where. So today I will talk about what is the impact of reduced soil microbial diversity on the plant productivity and the associated microbiome related to the seeds. So one of the most basic hypotheses in microbial ecology that we talk about is everything is everywhere but environment selects. It tells us about the immense potential of the microbes to spread and go everywhere, and they are everywhere. But who will survive in a specific environment? It depends on that environment, and who can thrive in a specific environment depends on the adaptations of these microbes. So this hypothesis was given by Beckings in 1904, and we're still working with this kind of hypothesis. So, like every beautiful thing on this earth, we humans, we like make mess of everything. And this is what we are doing now. And I'm not saying anything about Donald Trump, Simon. So, but still people say they don't believe that there is something called climate change. But I want to ask you guys whether you believe there is some changes or climate change going around us or not. If you believe, then it makes, we will be more relevant to you while I'm presenting this talk. So one of the studies done by my advisor, Dr. Max Colton, in salt marshes, and he says that when you have an increase in the temperature, you see a decrease in the richness of the microbial species. That means you don't have enough microbial species inside. And you see the same pattern on the diversity. Another study that we done recently in our lab, it says that when you have 3% of microplastic inside the soil, you lose 25% of the prokaryotic richness. That means 25% of the bacterial species are gone. And what is the impact? That we don't have enough seed production. That is directly linked with the seed production. And we also see that there is not enough ecosystem functionality in the ecosystem. So we agree on one point here till now, that due to all the events going around us, we see there's a decline in the microbial diversity. And these microbes, for example, they are the main source for the plants who are growing inside the soils. So soil, it contains a lot of microbes, and these microbes, they interact with the rhizosphere, and from rhizosphere they go to other plant parts, like in the last speaker, Tingting was telling. So from soil they go to rhizosphere and then to other plant parts, and finally ending up in the seeds. And these microbes, they help plant, like there are a lot of studies that shows like they help plant in the abiotic and biotic stress managements. So there are a lot of functions they can help in nutrient acquisition, they can help in all biotic stress or abiotic stress managements. So till now, we know that there is a reduction in the microbial diversity, but we want to see what will happen when you have reduction in microbial diversity on ecosystem functionality, plant productivity, seed-associated microbiota. And we hypothesize that the decline will have an impact on ecosystem functionality, productivity of the plants, and the seed-associated microbiota. But the seed endophytes, they will still maintain their functional repertoire because of the evolutionary relationship between the microbes or endophytic microbes with the plant. To address this, what we did, we took a green foxtail millet, Steria viridis, plant which is, and the relatives of this plant is, they are food for approximately 1/3 of the human population, specifically in Asia and in Africa. And we used this plant. Along with this, what we did, we took the soil, original soil from the Negev desert. We took this soil for gamma radiation, and we kill all the microbes inside. So we'll, in place of calling it as sterile, let's call it as irradiated soil, because many of the microbes, they can still survive the gamma radiations. We mixed these soils both in proportions, and we prepared a gradient of microbial diversity. And from this time, from now onwards, we call original soil as OS, and dilutions like D1, D3, D6, and sterile soil or irradiated soil. So what happens when we put gamma radiations, we also reduces the microbial biomass inside the soil. So we only wanted one variable, and the variable should be the microbial diversity, not the microbial biomass. Because many of the studies, if you have attended the morning session of Yakov studies, many studies by Professor Yakov is showing that there is a very clear evidence that the microbial biomass have an impact on the plants. So we only wanted to have one variable, that is microbial diversity. So what we did, we let this soil sit for some time, till the time the microbial biomass becomes very similar. And the time was, like we took samples every 14 days. So after 42 days, we see that the microbial biomass become very similar in all the soils. At this time, we put the seeds inside this soil, and we let the plant grow for whole life cycle. And then I will show you what is happening with the plant. So first we need to show there is a gradient of microbial diversity. So once we did sequencing of 16 sequencing from these soils, we can see, so I will show you like whole temporal dynamics, what is happening during the incubation, but I think you can focus only on the T42. That is the final time point. So you can see that there's a clear gradient of microbial diversity. It's not 1-to-1, we agree, but there is a gradient. And we can see this same gradient in the composition of the microbes. Interesting thing is, when you are looking on the reduced microbial diversity soils, the spore-forming bacterial genera, they are increasing. If you will see, they are, after 14 days, they were approximately 80% of the total abundance of the soil. But they are stabilizing around 60 to 65% after 42 days of incubation. And this is the relative abundance like statistics. We are also looking on the respiration, the energy demand of the system. So the energy demand is like we are looking on the basal rate of respiration of the microbes. And after 42 days, as I said, like when it becomes very similar, like the microbial biomass, so we are looking with different approaches that whether they become similar in overall activity or not. So this is one of the approaches. And we can say that after 42 days, they become almost or very similar in their basal rate of respiration. Similarly, microbial biomass, we can see after 42 days of incubation that they are very similar. So FDA enzyme activity, it tells us about the overall microbial functionality. And we can see from this data that after 42 days of incubation, the FDA enzyme activity becomes very similar. But from overall, if you look, it will seem like everything is coming back to the normal, but when you're going and looking on specific things or specific activities, you will see that things have changed. For example, if you look on, if you are providing them with different carbon sources, here we provided in the form of yeast extract, so they are not coming back to the same like the original soil. That means they can't utilize all the carbon sources that we're providing them. Similarly, another way of looking at this thing is we provide like 31 different carbon sources with the help of ecoplates, and when we want to check whether they can use the same or what kind of pattern they form while using the carbon. So we can see that the communities from original soil, they can use a lot of carbon sources, but reduced microbial diversity soils can't use it. So I have limited time, so I need to go a little bit faster. So this is phenol oxidase activity. It is kind of a proxy for the carbon cycling. So if you will see that after 42 days, the phenol oxidase activity is not coming back to the normal like that of the original soil. That means there are some changes in the carbon cycling of the soil. So urease activity is kind of a proxy for nitrogen cycling, and after 42 days, we can see that they are also not coming back to the normal like that of original soil. And same pattern we also see when we're looking on 16S data from the same soils. So the microbes which have the urease gene, they are also not coming back to the normal. Like if you can see here, Penibacillus and Pseudomonas, they are having differential abundance. Also, when you look on the gene expression, like the total gene abundance and urease activity, it's also being decoupled in the reduced microbial diversity. Only in original soil, after 42 days, we can see is still correlated. But under reduced microbial diversity, the activity of urease and urease enzyme is decoupled. So at this point of time, we are putting the seeds inside the soil, and we wanted to have look what is going on with the plants. So it's something like this. Initially, for 2 weeks, there were no changes. Everything looks very same, and I was so stressed, I thought like maybe I made some mess and something is going to happen. But things start to change after 3rd week. So you can understand it like this way, like till 18, our parents, they support us. But things start to take a different course when you have your own independent life. Similarly with the plants, after 18 days, they start to change. And you can see the plants after 3rd week, they already have inflorescence when they are going under reduced microbial diversity. Similarly, maybe they want to finish the life cycle very fast and want to go in a dormant stage in the form of the seeds. After 5 weeks, we can see a very clear pattern also in the plant growth, that plants growing under original soil are looking healthy and happy, and plants growing under reduced microbial diversity are having very short stature and also plant height. Similarly, when look on the stress indicators, so plants, they stress, when you don't have enough microbes inside the soil, they take it as a stress. And we can see from MDA and anthocyanin content here that there is increasing inside reduced microbial diversity plant. And you can see the plants here that are growing under original soil, they are just normal without any anthocyanin content inside them. But plants growing under reduced microbial diversity, they have clear anthocyanin deposition inside stem and the leaves. So the thing is, it's not only changing above ground level. If you look on below ground level, we can see clearly that the roots are also being affected. From here you can see that the same pattern, that roots which are growing under original soil, they have more root volume, root biomass, and everything higher than comparison to plants growing under reduced microbial diversity. But, okay. So I want to stress here specifically on the seed biomass. So if you don't have enough microbes inside the soil, so I don't think the future is very bright. We will not have enough foods or enough food to feed the people. So if you see here the seed biomass per plant, we can see that it's almost reduced to the half. So plants are not producing enough. But interesting thing comes now when you're looking on the seed nitrogen content and the seed nutrient content. So the plant is not having enough nitrogen inside them, not enough carbon inside them, but when it's coming to the seed, they are giving all the resources to the seed. And we can see it in the form of carbon and nitrogen. We don't know about the quality yet, but if we look on total nitrogen and carbon content, we can see that it's very much similar. Even the seed biomass is less, but whatever mature seeds are there, they have almost equal content of nitrogen and carbon. But if you look more deeply into the metabolites or non-metabolites of the seed, we can see that they are different. And specifically in amino acids, you can see that the seeds which are growing under original soil, they have more amino acids in comparison to that of the plants growing under D6. That means there is a changes in the quality. Okay. Let's have a look on the seed microbiome, what is going with the microbiota inside them. In Rajasthan, we can see there's a clear gradient like that of the soil microbial diversity. But when it comes to the seed, it's quite overlapping, and the plants are providing them with the very similar kind of microbial communities. You can see from here that seeds are having almost very similar kind of taxonomical phyla inside them, but they change in their abundances. It's a last slide. So we want to have look on what kind of, we see that something is becoming very similar, but what are the ecological processes that are governing this kind of assembly? So if you look here, that inside the rhizosphere is kind of more stochastic processes like drift or dispersal that are more kind of dominant. So this line, it tells us about if you have, if the value is above 0.05, that means more stochastic processes are more dominant. And if you have below this one, so more deterministic processes are more governing the processes. So here we can see that in the seed, it's more kind of deterministic processes that are governing the process of microbial assembly. So we can say till now that reduction in microbial diversity have an impact on carbon and nitrogen cycling, and in turn, overall ecosystem functioning. We can also say that reduction in microbial diversity negatively impacts the plant productivity and also being perceived as a stress. We can also say that plants are equipping seeds with almost similar kind of endophytic communities inside them, inside the seeds, and deterministic processes are governing the assembly of the seeds. Thank you.
Thank you, Uttam. We have time for questions?
Thank you. So, do you identify that the thing that the plant is always going to bring more resources to the seeds? Does it happen with all of the plants, and does it happen instantly?
It's kind of a common understanding that the parent plant always prefer to give whatever resources they have to the next generation, because that's the future that they are looking at. And that's kind of survival strategy for the plant. So in our case, we can see clearly that they are trying to give it, but I can't say about all the species yet.
Thank you, very nice talk. So what do you think? Is it that there is a bad actor in the microbiome that is now more abundant in the low diversity, and this is why the plant is suffering, or is a good guy missing?
Thank you, Omri, for such a nice question. So the thing is, we are not looking on, like in this perspective first for now, but the thing is, if I show you the spore forming bacteria, they are increasing a lot. So that means you don't have enough microbes which can help the plant to grow, because you need microbiota for nutrient acquisition. And these soils, Negev desert soils, they're already poor in the nutrient. So if you don't have those microbes which can help the plant to survive or to grow more, I think that is missing part here. We don't see any kind of, any bad guy being more active.
So, thank you for the talk. So I want to ask, you think the diversity is more important or abundance?
So, because my talk was focusing mainly on diversity, but as abundance, it depends on, you're talking about the microbial biomass, or specific microbes. So you mean xenon diversity? That's I mean. Is that mean xenon diversity? Because when you check diversity along with the abundance, that's xenon diversity. So
I mean, like now is about like is the quality, meaning like the abundance of single bacteria, like also diversity or abundance of single bacteria is more.
I think more diverse is much better.
Do you think like functions is like more important rather than like only diverse?
So the thing is, if I will put it this way. So there is the term called functional redundancy. So if you have more diversity, so if anybody is lost also, there is somebody who can do the work. So that means if you have more diversity, you can have more functional redundancy. So it's not about more abundance, it's more about the functions that can be done by somebody.
Thank you, Tam. I liked how this session developed, and I hope you did too. Thank you all for being here, and enjoy the rest of your evening. Thank you.